MedCram.com.
Welcome to another MedCram COVID-19 update. So this is probably the study that a lot of us have been looking forward to ever since the pandemic and these vaccines have come out. Imagine if you could test the immune system and its reaction to the Moderna vaccine, the Pfizer vaccine, the Janssen pharmaceutical vaccine, and the Novavax vaccine in the same population at the same time using tests on the same platform in the same lab. So you could eliminate all of those variables and actually compare what effects these vaccines have on the immune system. And then not only that, but after 6 months, be able to compare that to the effect of the natural infection of SARS-CoV-2 in the same population at 6 months. Well, this is exactly what Dr. Weisskopf and Dr. Crotty at the La Jolla Institute for Immunology have done, teaming up with Dr. Setti, who's also faculty at the University of California, San Diego. And so what I want to do is go over the results of this study, and this is just now in publication in the prestigious journal Cell, titled Humoral and Cellular Immune Memory to 4 COVID-19 Vaccines. And for those of you who want to cut to the chase, at least we can take a look at this. We are looking at the Moderna vaccine here in red. We're looking at the Pfizer vaccine here in blue. We're looking at the Janssen or the Johnson & Johnson pharmaceutical vaccine here in green. And yes, Novavax vaccine here in purple. And then, of course, the SARS-CoV-2 infected people, which we'll talk about more in details here in yellow. And at 6 months of immunity, 6 months after this initial vaccination and or infection, if you look at binding immunoglobulins, you can see the profile here with this dotted line being the lower limit of quantification. If we're looking at neutralization of a pseudovirus of SARS-CoV-2, you can see here the profile among those. And here with CD4, with the circulating T follicular helper cells. You can see that here as well. CD8, and then of course the all-important memory B cells and the profile that is seen 6 months after either vaccination or infection with SARS-CoV-2. Now remember that this was done in San Diego, and so they did not have access to people who were vaccinated with the AstraZeneca vaccine, for instance, or Sinopharm or Sputnik. So these are ones that are specifically authorized for use in the United States. So stick with us and let's get into the details of this study, which I think are really interesting. And of course, we'll put a link in the description below to this article so you can read it for yourself. So again, what they did in this study was they looked at 30 people who had the Moderna vaccine, 30 people who had the Pfizer-BioNTech vaccine, 30 people that had the Janssen vaccine, and 12 in this case that had the Novavax. We'll talk more about exactly that. As you can see, the female to male ratio in all of these groups was about the same, a little bit more equitable in the Novavax vaccine, and also slightly younger age group in that Novavax vaccine group. In terms of race or ethnicity, they tried to keep that also pretty similar across the spectrum. What's really important to understand is the exclusions as well. Anybody with neutralizing antibodies or nucleocapsid antibodies, which would signal that the patient has been infected previously with SARS-CoV-2, were excluded from the study. They wanted people who had never been exposed before to either the virus or the vaccine. Also to that degree, because it's not obvious sometimes if you've been infected. If they had any peaking of the immune results after the first dose, this is usually a sign that there's been a previous infection and that you're getting hybrid immunity. Those people were also excluded from the analysis. And of course, if anyone went on and got a third shot, or a second shot in the case of the Janssen pharmaceutical vaccine, they were excluded from the study. So they're just looking at people with 2 shots for the mRNA vaccine, 1 shot with the Janssen pharmaceutical, and 2 shots with the Novavax. The point of this, again, is to look at the different platforms. Obviously, the Moderna and the Pfizer vaccines are both mRNA vaccines. And remember what that means. That means that an mRNA molecule goes into the cell of the human body, just like it does in the natural infection. And that causes the ribosomes to make the spike protein, which then causes the immune response. That is as opposed to the Johnson & Johnson or the Janssen pharmaceutical vaccine, which is an adenovirus vector, which infects the cell and actually has a DNA molecule, which is then transcribed into messenger RNA and then finally into the spike protein. So that's a little bit of a different platform. And then, of course, the Novavax, which is a more traditional type of vaccine where it's simply the protein that gets injected into the body and that makes the immune response. So interesting to look at these three types of platforms and see how they are different. Again, we're not here trying to say which vaccine is better, because remember that vaccines not only have benefits on the benefit side, but they also have risks on the side effect side. And so this is not addressing any of the issues with side effects or risks. This is coming out of an immunologist's laboratory, which is looking at the effects on the immune system. And so obviously we're going to get a lot of good data on that aspect. Just keep in mind that, for instance, while the mRNA vaccines have a low incidence of myocarditis, you wouldn't necessarily see that in the Johnson & Johnson. And conversely, as we all remember a number of years ago when we first started, that the campaign for vaccination for the Johnson & Johnson vaccine had to be paused because of a concern for blood clots, which were not seen in the mRNA. So the point of all this is that we're looking at one aspect here. We're looking at the benefits, we're looking at the efficacy of vaccines and their ability to protect against infection and also disease in SARS-CoV-2. So we've showed you how they came about getting subjects for the vaccination in the 4 different groups. The next question is how did they get the subjects in terms of comparison for SARS-CoV-2 infection? That comes from another research article that was published by the same group, as you can see, out of the same La Jolla Institute for Immunology. It was published in 2021. They received and were able to get the data specifically 6 months after infection. And here's a quote from that article specifically talking about that cohort that's going to be looked at in this particular current paper. And they say, quote, all of the convalescent donors experienced mild illness, defined as patients with a SARS-CoV-2 positive test who have never been hospitalized. Seropositivity against SARS-CoV-2 was confirmed by enzyme-linked immunosorbent assay, or ELISA, as described below. At the time of enrollment, all COVID-19 convalescent donors provided informed consent to participate in the present and future studies. So in the current research that we're going to look at, we are going to be looking at different points along the way when these subjects got their vaccine. That's not going to be the situation when we're looking at these patients in this study that got COVID-19. We're only looking at a snapshot in time at 6 months. And that's going to be important to understand because when we throw up these numbers and graphs in this current study, anytime we're comparing them to people who have had COVID-19 through SARS-CoV-2, it's going to be a snapshot at around 6 months, which of course is important because we want to know how much immunity do subjects have at 6 months. So I just want you to be aware of that. So let's take a look now at our vaccine subjects. And so what you can see here is we've got the Moderna vaccine here in red again, Pfizer in blue, Janssen in green, and Novavax in purple. The time periods that we're going to be looking at, for instance, this is going to be T1, this is going to be T2, this is going to be T3, this is going to be T4, and this is going to be T5. And all of the data that we're going to get at T1 is going to be prior to vaccination. That's what the blood was like right before the first dose. And so T1 in all of these first 3 are going to be baseline examinations. And as we go through T2, T3, T4, T5, and this is around 6 months. And this is where we are going to be using these data points here to compare against the SARS-CoV-2 infections. You may notice something here. We have T1 through T5 for the Moderna, for the Pfizer, and for the Johnson & Johnson or the Janssen pharmaceutical vaccines, but we do not have it for the Novavax. And that's important to understand why that's the case. The reason why this is the case, now listen very carefully, is because when they did the Novavax randomized trials, the phase 3 trials, they did not want to leave people out. They wanted to make sure that every single one of them was vaccinated eventually. And so what they had to do was they had to do 2 sets of 2 vaccinations. People were randomized so that the first series was the placebo and the second series was the actual vaccination. And the second randomization was that the first dose was the vaccination and the second was the placebo. And so because of that, we can only look at T4 and T5, knowing that this is what things looked like after vaccination at 6 months. So now realize they did not receive any doses here. They only received placebos, as it's mentioned here. And so as a result of that, we could not be drawing blood here and here in those patients because we wouldn't be able to keep them blinded, because then they would know what group they were in. So as it turns out, what we can only look at in the Novavax is T4 and T5. And this is a figure from the article that simply just codifies what I was just saying here in graphic format. So let's look at the data. And the first sets of data here that we're looking at is specifically the spike IgG. So this is the antibody that's circulating around in the body that is going to bind to the IgG. And just a point here of note is that the virus that we're looking at here is very early on in the pandemic. We're not looking at variants. We're not looking at the Delta variant or the Alpha or even Omicron. We are looking at the native virus. So a lot of this may not be applicable to today in terms of the Omicron variant, but what this does give us information on specifically is how these different platforms, whether it's an mRNA-directed vaccine, whether it's an adenovirus vector, or whether it's a traditional protein platform vaccination, how do they affect the immune system in a way that protects against SARS-CoV-2? So again, we're looking at antibodies against the entire spike protein. This is an IgG, which means it usually hangs around. The other thing that I want you to notice here is this scale is not linear. It is logarithmic. And so at any data point, the number of hash marks that you cross is the number fold of difference. So if you cross 2 hash marks, that's twice the level. Between 10 to the 2 and 10 to the 3, that's 10 spikes, so that's 10-fold increase. And so what you can see here is at baseline, which is very low here for the Moderna vaccine, we had a very high rise here in the titer of anti-spike IgG. And then at around T3, there is a pretty precipitous decline. This doesn't look like a precipitous decline, again, because it's logarithmic. But if you notice here, we are crossing at least 3 to 4 hash marks, which means that is a 3 to 4-fold drop in the levels of antibodies that we're seeing here. But also notice across the board that the mRNA vaccine, specifically the Moderna and the Pfizer vaccines, had the highest titers. The Johnson & Johnson did not see very high titers, but you can see that it was actually pretty consistent across the board. With the Novavax, this is the traditional protein type of platform for vaccination. There was an increase here at T4, so again, we're going to compare it to all of these T4s. But there was still a decline even after some period of time, although it was a decline numerically, but it was not statistically significant. Now, what we're seeing here on the right-hand side is what's going on at 6 months. This is with just 2 shots for the mRNA and just 1 shot for the Johnson & Johnson. And so again, in red, we have Moderna. In blue, we have Pfizer. Green is Janssen. The purple is Novavax. And here, yellow is the infected SARS-CoV-2 cohort that we talked about previously. And I think that the key here to understand is that, for the most part, there was circulating antibodies in all of these groups. In all of these graphs, this dotted line here represents the lower limit of quantification of the assay. And so there was 1 individual in that cohort, in the Novavax cohort, that had undetectable anti-spike IgG antibodies. And if you want to look at the means, you can see here how it was different across the board. And again, remember, each one of these hash marks is a fold increase or decrease depending on the direction that you're going. So now what we're going to do is we're going to look at the receptor binding domain, which is a very specific portion of the spike protein. It's the very tip, if you will. And what you see here is things that are very, very similar that we saw before. Again, here we look at the lower limit of quantification. And here we have 2 individuals that went below that, and another 2 in the infected group that went below that as well. This kind of goes along with what I've heard Dr. Crotty mention before in terms of vaccination versus infection, and that is that vaccination typically gives a more consistent response, whereas infection may not be so consistent. And again, remember, with this cohort, none of these people went to the hospital. All of them had mild COVID-19, so we don't know what the response would be in those that were sick enough to be hospitalized. And if we look here, we can see that the Johnson & Johnson cohort, again, pretty tight, but also the mean was almost an order of magnitude lower in terms of receptor binding domain IgG antibodies. Okay, what we're looking at here is pseudovirus neutralization. This is a universal type of test that we do all over the world that checks to see what the antibody titers are in a person's serum that can neutralize a viral particle that's coated with the SARS-CoV-2 spike protein. That is also very interesting to look at here. You can see Moderna, very high titers, which are good. And here you can see the Pfizer. The Johnson & Johnson Janssen Pharmaceutical vaccine is not as high, although we do see pretty high titers here with the Novavax. And again, you can see here in terms of the pseudovirus neutralization titer what the results were. And you can see the difference here between Moderna and the SARS-CoV-2 infection was very highly statistically significant in terms of neutralization titer. You can also see that there is a larger splay in terms of response in the natural infection for those that had mild infection. It's even larger here in the Novavax vaccine, again, with 2 individuals that were below the lower limit of quantification. So all this caused the authors to write in their results section that antibody titers at 6 months were compared to SARS-CoV-2 infected subjects who were enrolled for a previously reported study. The previously infected individuals were selected randomly. Recipients of the mRNA vaccine, so that's both the Moderna and Pfizer, had a 4.5-fold higher spike IgG, a 6.4-fold higher RBD IgG, and a 3.4-fold higher neutralizing antibody titers compared to previously infected subjects. Antibody titers from Novavax recipients also trended higher than SARS-CoV-2 infected subjects. Antibody titers from the Johnson & Johnson were similar to titers from SARS-CoV-2 infected subjects. Okay, so that's interesting to know in terms of antibodies, but we all know, and we've all learned here very well over the last couple of years, that immunity and the way you stay out of the hospital is more than just antibody response. You also need to look at T cells and memory B cells. So let's take a look at that now. So here we're looking at something called spike-specific CD4 T cell percents. They had different assays that they used at La Jolla. One of them was this AIM Plus assay, but there are other assays that they used that I'm not showing because they had the same results, but feel free to look up the paper and see those other assays. This is very representative of what we saw. Again, we're looking at a logarithmic scale. It's important for you to understand that. And you can see here that it's very important. This is something that they were looking at very early in the pandemic, is whether or not these vaccines were able to elicit a T cell response. Because if you can elicit a T cell response, then that means you can get much further and much more robust cell-mediated immunity. And you can see here that we had a very good, robust response with Moderna, also with the Pfizer vaccine, and really also with the Johnson & Johnson vaccine as well. And very happily with Novavax. So Novavax had a very good T cell CD4 response as well. In fact, arguably it had one of the highest in terms of after 6 months. Again, we're looking here at 6 months. What is the T cell response? What is the CD4 T cell response after 6 months? And we've got Moderna, we have Pfizer, we have Johnson & Johnson, we have Novavax, which actually had a very tight and elevated response in terms of CD4 T cells, all in comparison with those that were, again, naturally infected with SARS-CoV-2, but did not have to go to the hospital. In the study, they looked at spike-specific CD8 positive T cells, although they did not do a comparison that I could find to those that were naturally infected. You can see here robust responses pretty much all across the board. However, what was interesting is at the T4 period of time, it was unclear why that was such a low response in that situation. But at T5, there was an increase above the lower limit of quantification. So a lot of time and effort has gone into looking at something called memory B cells. That's really important. We're specifically looking at the receptor binding domain, which is where the SARS-CoV-2 virus binds to the ACE2 receptor. And in all of these cases here, you can see from T4 to T5 all the way across that there was a robust response, which is good. It means that at least in terms of mounting a response down the road, if someone becomes infected again and again, that there's going to be a response that keeps people out of the hospital with cell-mediated immunity. If we compare those again with the vaccine cohorts and those that were infected with SARS-CoV-2, we can see that generally speaking, all of these are good responses. Again, these are at 6 months. And if you wanted a better look at that, again, we're looking at spike memory B cells. And we're looking at a logarithmic scale. And so any kind of deviation here is a fold difference. And so the authors of this study state that the longitudinal analysis of T cell, B cell, and antibody response to 4 different COVID-19 vaccines in humans, representing 3 different vaccine technologies, identifies different quantities and quality of CD4 positive T cell, CD8 positive T cell, and antibody response elicited by the Moderna vaccine, the Pfizer vaccine, the Janssen vaccine, and the Novavax vaccine. Antibody levels decline, but memory T cells and B cells were comparatively stable. So in terms of limitations of the study, they say here that they did not evaluate the recognition of variants. They also did not test, as we mentioned, AstraZeneca, CoronaVac, Sinopharm, and Sputnik because those were not available in the United States. In terms of funding for the project, it came from the National Institute of Allergy and Infectious Diseases, the National Institutes of Health, and the Department of Health and Human Services. And you can also see the declaration of interest as well. Again, we'll put the link to the article in the description below, and also join us at medcram.com for over 60 hours of continuing medical education and review of medical topics. Thanks for joining us.